Appreciate the opportunity to get to talk to you today. Brad Niles, CEO of ARIZ Precision Medicine. We are the PRDM oncogene targeting company. We use that tagline. I'll explain a little further into it, but it's about finding the root cause of the cancer, targeting the change that happens in the cancerization of the cell through this family of genes and proteins known as the PRDMs. Our goal really is to make a difference in the lives of the patients, and in terms of a therapeutic option that is killing the cancer but not harming the healthy cells. The real need for that is in the non-small cell lung cancer space, showing here survival rates, diagnosis of stage 3 and stage 4, which is where the lung cancer is most commonly diagnosed. We're talking below 20%. 5-year survival rate. So these patients have a real need for new therapeutic options where we can diagnose earlier and treat better. At a glance, we are developing this portfolio of multiple candidates targeting a previously undruggable gene known as the PRDMs through an innovative new approach. We've used that to demonstrate more than 90% cell killing in an in vitro cell culture model of these lung cancer cells. As well as an animal model where we got 80% reduction overall in tumor growth and 24% better than standard of care chemotherapy. We've been recognized by a number of groups, including being named the Buzz of Bio winner at the Bio International Conference a few years ago. We're recognized by J&J and also were accepted to the prestigious CLSI FAST advisory program. I've assembled a great team to help us get through to including regulatory and clinical development to get to the point of human phase 1 trials, which is the point in which we would like to license this product to a pharmaceutical partner. Our business model is in this early stage development, getting the licensing deals that commonly occur in the pharma business and taking those large upfronts to further develop our additional products. Our scientific advisory team is headlined by Michael Shepard. You may know the name as one of the inventors of the blockbuster breast cancer drug Herceptin for Genentech a few years back that made an amazing difference in the lives of breast cancer patients. Our target is this in lung cancer is this gene known as PRDM2. And the reason we have chosen this is been shown a couple of years ago in this landmark paper demonstrating the role of modulation of PRDM2 by the tobacco carcinogen NNK. So what this boils down to is smoking cigarettes leads to a change in our gene, and this change leads to cancerization. So you can see here as early as 3 days, you're getting initial changes of this gene. With continued smoking, this develops into a tumor, and you continue to have this disruption of the gene within the tumor itself, demonstrating a real causal effect of smoking leading to cancer through this particular target. Of course, have a lot more information on the target itself that I'm happy to share with you. But what is really interesting is that if you look at human tumor patient samples, a very high percentage of these patients actually have a disruption in our gene. And you compare this with a product like Herceptin, there's a massive blockbuster drug for Genentech, we're hitting even a higher percentage of that. So it's a very large number within a very large market. Our approach is, actually I'll get into it a little bit later at the end. We use this siRNA approach to go through and target a truncated version of the isoform that exists only in the cancer cell. In this way, we're able to knock down what we call inhibit the inhibitor concept, knock down the target. This increases the level of the tumor suppressor gene, which in itself kills the cell. We've been very effective at showing demonstration of cell killing in vitro. Here you can see in the second set of bars over, we're getting more than 90% cell killing. We're also very effective in an animal model as well. You can see here at the red line down at the bottom, we're getting more than 80% tumor growth inhibition through specific target knockdown. We have a product pipeline in place for lung cancer and some other products as well. We're looking to raise $1 million. We've got $650,000 of that already committed for research purposes on additional steps towards an IND and eventually moving forward with a Series A. Our valuation is $10 million. We're raising off a convertible note.